For 13.2, it'll be about photosynthetic pigment. Now, whenever I say photosynthetic pigment, I know chlorophyll is what comes to your mind. Yes, that is there, but like I said, A levels are more detailed, no? So you have a little bit more. Right. So I'm going to break down the chlorophyll like this. So first of all, we have chlorophylls. We actually have types of chlorophylls. Now we have two main types of photosynthetic pigments, and we also have two types of chlorophylls. I'm going to start off with the two types of photosynthetic pigments. I'm going to use a pale orange for the next one. So here we have chlorophylls, and then we have a set of pigments known as carotenoids. Carotenoids. Hold on, let me mute my phone. I really thought I muted my phone. Okay, sorry. Right, and then we have carotenoids. Just a small thing to keep in mind. So like a small segment to anchor you to this. You know how carrots are orange? That's because of beta carotene pigments. We'll talk about that. Now, when it comes to chlorophyll, is not much of a big deal. We have chlorophyll A, sometimes referred to as chlorophyll alpha, but it's not really that predominant. And then you have chlorophyll B, sometimes referred to as chlorophyll beta. Don't worry about that, just call them A and B. Okay. And then when it comes to carotenoids, we have also two types carotenoid. Now, it's not carotenoid A and carotenoid B. I would also have loved it if it was like that, but it's like this. Carotene is the first carotenoid pigment. This is sometimes referred to as beta carotene, but we'll just call it carotene for now. And then the next pigment, I have always thought of this pigment as a really good name for a band. You know, like the Beatles and stuff. I'm not sure, I might be losing my mind, ADHD mind, but I think xanthophyll is such a good name for a band. Don't you think so? Leave your suggestions in the comments, I guess. Carotene, and then you've got xanthophyll. Right? Now, I will be specifically talking about the color of the pigment. So I'm going to just make a small column here. Pigment color. Chlorophyll A is a yellow greenish pigment. Okay. Chlorophyll B is more of a blue greenish pigment. Chlorophyll B is more of a blue greenish pigment. Carotene is actually a, like I said, remember, carrots are orange because of carotene. So that is simply an orange pigment. Xanthophyll is a little bit below orange. That means that xanthophyll is actually a yellow type of pigment. Orange, and then we have a yellow type of pigment. Now, this is a little tiny bit of excess information, but a little bit of excess information never hurt, right? Now, I'm going to tell you how exactly these pigments are layered up. So in a photosystem, that's what you call when all these photopigments come together, when all these photosynthetic pigments come together, we call them a photosystem. We'll talk about photosystems in the stages of photosynthesis. Don't worry about that too much. But I'm just going to show you a very simple photosystem. Typically, this is how these pigments are laid out. So if you didn't count, there's 4 pigments. We have 4 pigments. It's like this. 1, 2, 3 on top. Oops. 1, 2, 3 on top. Like so. And then you have one primary pigment at the bottom. Now let's specifically assign what is what. So here this, like I said, this is our primary pigment. This is our primary pigment. And this is actually sometimes also referred to as the reaction center. Why? That's because it's like this. You know, sunlight comes and hits the topmost pigment. right here. And then from that, energy is transferred to that, to that, to that. And that's the final place energy can go. No? So that's where the reactions happen. So that's why we typically call it the reaction center, and that is why it is referred to as the primary pigment. So the primary pigment is actually alpha chlorophyll. So chlorophyll A. That's our primary pigment. Above that, we have chlorophyll B. Nice, simple. We've got chlorophyll B, carotene, and the topmost pigment is actually xanthophyll. So xanthophyll is what I really hope I'm pronouncing it right, even though it's a band name. Xanthophyll is the one the one that's going to get hit with sunlight. That's the direct pigment that's in contact with light. It transfers its energy to carotene, carotene to chlorophyll B, onwards to chlorophyll A, like so. This is the primary main pigment. No? So these pigments are typically referred to as the accessory pigments. The accessory pigments, no big deal. Now we have to talk about something known as a absorption spectrum. Something known as an absorption spectrum. Now we know that all of these pigments are capable of absorbing light. No, there's no doubt about that. No, but different pigments are capable of absorbing light in different wavelengths. So there's a distinction in that. We have to be aware of that. And typically what is given to you is something known as an absorption spectrum or as absorption graph. This is simply a graph of all the pigments that are going to absorb light and of what wavelength. So on the y-axis, we have the amount of light absorbed. And then here we will be talking about the wavelength of light, typically in nanometers. Okay, so here we have about 400 nanometer light. That would be around blue, basically. So blue, and then you have the 700 nanometer range. That would be reddish, so blue to red. Okay, now let's observe how these pigments are going to behave. I'm going to start off with something that goes something like this. And there's a huge dip, and boom, and then there's a rise here, and then there's a dip. That's the first one. Now you don't have to memorize the shapes at all. You just have to identify them if this spectrum is provided to you. You don't have to draw. I'm going to repeat that. And then I'm going to draw a second pigment that's going to go a little bit like this. Its peak is about there. And again, it drops and it comes all the way. And there's a small peak here and then it drops and returns. One final thing. This has actually a very weird kind of shape. So boom, and it drops and there's nothing else. It just drops. Okay. So here we have illustrated 3 pigments. Now we know we have 4 photosynthetic pigments, 4 photosynthetic based pigments, but here we only have 3 pigments listed. All you have to do is know how to identify what they are. Now this shape, broom, and then this huge rise, that is chlorophyll A. Simple, straightforward, nothing. And then right next to him, this guy who rises about here and has a small peak, that would be chlorophyll B. That would be chlorophyll B. And these are, so this is simply speaking, this line is when combined. Carotene and xanthophyll, they're combined here, so you typically refer to them as the carotenoids. Right? The carotenoids. We can't really, it's not easy. Just by using these UV visible spectrophotometers, we can't really differentiate between carotene and xanthophyll. That's the thing. So chlorophyll A, B, and the third set of pigments, that's why I've said 3 pigments, the carotenoids, they show similar behavior. This is what is known as an absorption spectrum, sometimes also referred to as a UV spectrum. You don't have to draw, you just have to know how to identify each and every one of them. Okay, so if this is provided to you, you have to tell. So now let's just go through it a little bit. Do you see? Chlorophyll A is really good at absorbing light in the bluish violet type of region. Do you see this blue to red? No? It's really good. And chlorophyll A is also good at absorbing red light. Now, do you see? Carotenoids have no ability to absorb red light. See, it dips here, it dips around somewhere here. So blue, violet, something like that, dips around here. Chlorophyll A dips, but it rises over here. It's capable of absorbing red light as well. That is why we need different types of photosynthetic pigments to maximize light absorption in different wavelengths. Now, these are colors to us. Different wavelengths mean colors to us as humans. But when it comes to plants, plants don't see colors. They're talking about wavelengths. Different wavelengths. You know, the sun is, which is the main source of energy when it comes to the entire earth and the ecosystem, it emits different wavelengths of light in its emissions. Plants have to be able to utilize all these different forms of wavelengths to maximize their food production, or in other terms, to maximize the rate of photosynthesis. That's the deal. Now, along with this, now just next to it, I'm going to bring something else in as well. This is a very similar graph to that, but this is not an absorption spectrum. This is what we refer to as the action spectrum. Here we're talking about the rate of photosynthesis. Now this is known as an absorption spectrum because we're measuring the amount of light absorbed. Here we are straight away measuring the rate of photosynthesis, and this graph is linked to the absorption spectrum. But how? I do not know why this axis is not labeled. Might have missed it. So rate of photosynthesis, that is wavelength in nanometers. And this, now this is going to give you a better idea. So do you see violet, blue, green, red? That is how it goes from left to right. Do you see? Here we have a lot of activity around this area. That's why we have a peak here. Now, do you see everything drops now when it comes to the middle? So there's not really that much absorption in the greenish area. Now, that is a very good analogy. I mean, not an analogy, that's the truth. But that is why plants appear green. You know, any object, whatever color it's going to reflect, is the color of the object. No. So if you have a red color ring, that means that that ring is going to reflect the red wavelength and absorb everything else. That is why plants are green. Plants do not absorb green. That's the deal. Plants mainly absorb blue and red. Plants reflect the color green. That is why plants appear green. That's the idea. And as you can see here, we have a lot of chlorophyll A activity. Therefore, the rate of photosynthesis is also high over there. Just to put this into your mind, here we are talking about absorption. How much light is absorbed. Here we're talking about the rate of photosynthesis. We are just directly linking this into an action spectrum. You are also not expected to draw the action spectrum at all in any form of way. It's just to give you an idea. Now, do you see here these pigments, and you have a high rate of photosynthesis. Here we've got these pigments, and there's a high rate of photosynthesis, like so. The final part when it comes to these photosynthetic pigments is a rather interesting one. We have to talk about pigment chromatography. Now, how exactly did scientists come up and say that we had 4 photosynthetic pigments? Of course, they conducted a research. That's how everything works in the scientific world. Now, when it comes to pigment chromatography, it's very simple. Now, you know what chromatography is from your ordinary levels and your chem right now. I will just simply give you. So distance, traveled by the pigment, I'll just call it the spot, and then you divide it by the distance traveled by the I'm sorry, my handwriting is going off, by solvent. Okay, so sometimes mobile phase over stationary phase, same thing. I will also write that down. So here we're talking about the mobile phase on top and the stationary phase on top, and you're going to get a ratio when you divide this by this, like so. So I'm just going to draw a sample chromatography paper. This part is not hard at all. Pay attention to the values I'm going to provide. That's going to help you a lot. Okay, that is our chromatography paper. When you do this experiment properly, you put the baseline below the solvent level, above the solvent level, and then you let the ink, not really the ink, but the photosynthetic pigment to move through the solvent front. You're going to observe something like this after some time. A spot here. And then you will have a spot just a tad bit above that. And then you will, I have no idea what that was. And then you will have another one. I think it was probably a bird or something here. And then one pigment goes all the way up until almost the edge, not the edge, but almost the edge. Okay, right. Now, what are the specific retention factor values? What are the specific RF values of these? This you have to remember, and I'll make it easier for you to remember. Okay, here this has traveled with a RF value of 0.95. This has traveled, I'll start with the small one, 0.45, and the above one is 0.65. And here this pigment has actually traveled 0.71. Okay, but now do you see all these things are multiples of 5? No, you don't have to be that specific. You can include little tricks like this to make it easier to remember stuff. So typically what I tell everybody to do is to remember it as 0.75. It's perfectly fine. It's 0.71. I will also write that down in the brackets, but you don't have to be that specific. You don't have to stress yourself that much over a pigment and an RF value. Just remember it as 0.75. Now, if you were following the color grading, you'll know that this is actually chlorophyll. What do you think? B. It's not chlorophyll A. You might think chlorophyll A is the starter, but no, this is chlorophyll A. Okay? Do not mess these two things up. The 0.45 traveler is chlorophyll B. Okay? 0.45 traveler is chlorophyll B, not A. All right. And then when it comes to 0.75, that is xanthophyll. And then you've got carotene, the final thing. Now, if you were following the color grading, you would know that I typically use green for chlorophyll A and pink for chlorophyll B. Here, I wanted to specifically focus on that. Here, this is actually the chlorophyll A pigment. Okay, so this is flipped. This is flipped, meaning I'm gonna write them in this other, color on top. This is actually chlorophyll B, 0.45, and this is chlorophyll A. I wanted to make that distinction clear. Okay, chlorophyll B on the bottom, chlorophyll A on the top, and that would be all for pigment chromatography and all these photosynthetic pigments as well.